we have time for a few questions for our speaker? Would anyone like to ask questions?
Yes.
I know you said not much is known about the vanadium or the iron containing.
Yeah.
I assume that people crystallize those already?
No.
But you know the sequences of those.
Oh yeah. Yeah. So the question was, I guess, what do we?
Similarities struck at the sequence level that would predict something.
Yes, asking questions about the relationship between these alternative vanadium and iron containing nitrogenases to the molybdenum nitrogenase. So at the sequence level, they're very similar. And so the idea that the main difference between them is that you have vanadium replacing molybdenum or iron replacing molybdenum, I think has to be a reasonably good representation of what's happening. It turns out that the proteins are very similar, and also then there's something like 20 genes that are required to fix nitrogen, of which maybe a dozen of those, so there's 3 that are the structural genes, there's some regulatory ones. Most of the other ones are involved in making the cofactor, not the P cluster, which is interesting. And those proteins are very similar in all these systems as well. So the enzymes involved in biosynthetic pathway are similar. And in some cases, the molybdenum enzyme may be used in one of these other systems as well. So it's an interesting problem. So you have these soil-living bacteria, and they'll contain these 3 nitrogenase systems, which ends up, like being 60 or 70 genes. And they're highly regulated, so if they're expressing molybdenum, if there's any molybdenum around, they will not express vanadium or iron. And actually demonstrating that you had alternative nitrogenases before, so you could sequence things, was really challenging because it's hard to avoid contamination, and these systems are really, if there's any molybdenum around, they'll use that. So I look at this and I say, molybdenum's pretty soluble, and it's not that unusual, and it's like if there's not enough molybdenum, you wonder how can there be that much iron. But maybe I would just give up and say, like, if there's not enough molybdenum, and then especially if there's not enough vanadium, it's hard to believe there'd be enough iron. So, but these are real survivors, and so they have plan A, B, and C, to eke out an existence. But I know that people have tried to crystallize these. Some of them have used our glove box. We weren't sort of a sabbatical visitor for someone at Caltech, someone else at Caltech tried it. They have additional subunits, and I think that sometimes they fall off, and so you have a heterogeneity problem. Yeah, it'd be an interesting thing to do. Yes?
Thinking about the iron molybdenum complex and looking at the symmetry of that, sandwiching of the sulfur, et cetera, I'm sure that's tantalizing to people. Have people done electronic structural modeling of that and tried to look at the charge distribution in that?
Yeah, so the question is, have people looked at the electronic structure and charge distribution and so on? Yes. One of the things I find is sort of an interesting commentary on things. So when we first proposed the structure of the cofactor, we didn't have this interstitial ligand. And so a number of calculations were done, and they all seemed perfectly fine. And then we discovered this atom in the middle, okay, which was our fault not seeing it the first time. And then people were unhappy that they'd been doing all these calculations and there wasn't iron in the middle. So there have been a lot of so these density functional theory methods have been used to try to model, and those generally come up with liking nitrogen in the middle. But I think there's a real question as to the overall oxidation state of the metal center, and there's some consensus models of what they should be, and in general the calculations seem to be consistent with that, but I think in order to do these calculations, generally, oftentimes they try to come up with charge neutral states, and so that limits the number of electrons and protons that can be there. And so I sort of have to wonder if they've how accurately been able to model this. Having said all that, I'm convinced that our ultimate understanding of the mechanism will only come from computational chemistry, that there are just too many intermediates and so on, that even my most delusional state of imagining how many intermediates we could try to characterize, if we could get one intermediate, that would be like amazing. And so I think ultimately computation will be required to fill in the step. And there have been a number of computational studies on the reduction mechanism, but they can't all be right. I mean, and I think that there's not enough experimental constraints to really guide the development of those models. Yes.
Make a comment and then a question. You've done probably our students a great disservice because they didn't say this guy from Caltech showed that mechanism for a reaction, therefore I can do that. Let me tell you, that's not true. And so you said that there was a large number of intermediates, and that's one of the limiting factors. I mean, so many complicated biochemical mechanisms have been teased apart by careful experimentation. What are the technical, philosophical difficulties with doing those kind of approaches with these things? For people that have done biochemistry, for example, for 1 year, have learned about how people have found intermediates or have, fed different feedstocks into enzymes, and so why can't we do that with this?
Yeah, that's a great question. I think it's got, in a way, it's embarrassing that you have a slow enzyme with dinitrin as the substrate. So, right, I mean, we're not talking like, 50 atoms or whatever. I mean, you have 2 atoms for the substrate, and it's a turnover is about a second. So you don't need, femtosecond time resolution to determine the structure. So it's pretty astonishing. It's actually shocking that there's no better insight into the mechanism. So why is this? So first off, I think that there's a sociological phenomenon, and that there are not many people working on this system because it's oxygen sensitive, and people don't want to work on oxygen sensitive systems biochemically. Inorganic chemists, no problem. Our stuff, if you expose it to oxygen, it just gets inactive. Their stuff, you expose it to oxygen, it catches on fire or whatever. So these are not nearly so. You can find and in the bioinorganic community, there are more people than you can shake a stick at doing iron-oxygen chemistry. But until maybe the last 5 or 10 years, there's been no one doing, say, even model chemistry on iron-nitrogen chemistry. So we have Jonas Peters, there's Pat Holland at Rochester, and so on. So it's starting to change a bit. Actually, Dick Schrock at MIT won the Nobel Prize for his work on metathesis. Also has been interested in nitrogen fixation. I mean, he believes in molybdenum, but I think he's helped raise the profile of mechanistic chemists. I think there was also, in the 70s, there was a huge emphasis on this problem during an earlier energy crisis, and that this was going to, something like 5% of the natural gas or a few percent of the Earth's energy consumption goes into fixing nitrogen. So if you could fix nitrogen more efficiently, then you could save energy. And so there was a lot of interest at that point to find if you could make cheaper catalysts. So my sense is now that that project, I think, ended without that interest ended probably in around 80s or so, because they weren't able to make any headway on the project, and but it created this sort of picture that this was a hard project. It also, I think the Haber-Bosch process on a per mole basis is the world's largest chemical industry. I mean, making ammonia. And so if you're going to have a chemical process that could replace the Haber-Bosch process, I mean, it has to be incredibly efficient. When you look at these catalysts, there's no way that this is going to happen. So anyway, I think a community got interested in it, then they got turned off from the project. There are few model chemists, there are actually few biochemists. It was dominated by a few schools, and Those were mostly headed by senior people who'd sort of divided up the field, and they've gradually moved on. And not so many younger people, again, moved in, I guess thinking that this was too hard of a problem. So Lance Seefeld at Utah State, Marcus Ribby at Irvine are some of the few, but again, it's starting to be populated. So but I think the problem was sort of split up into different pieces, and the people working on the mechanism were mainly spectroscopists. And so that depends on, having some isotopically labeled substrate and having it interact with some spin state of the protein where you could see signals. And one problem with a reaction that involves at least a minimum of 6 electron transfers, maybe, is that you'll end up getting a series of a big mixture of a mixed population of MoFi protein in different oxidation states. And so you don't have just a really pure form. Substrates don't bind to the MoFi protein as isolated. It has to be reduced by 2 electrons. That only takes the iron protein and ATP. So it's not something you can easily do in a crystal. Although we hope that sort of maybe radiation damage, we can get more highly reduced forms. So, I think it's a combination of things. I mean, I think if someone told me in 1992 that here we'd be almost 20 years later and that I would give a talk, I'd be happy to have all this stuff here. But a talk where we say we don't know anything more about the mechanism, I think the feeling was that within 5 years, this problem would be solved. And so it took me about 10 years to realize, geez, the problem's not solved. And, we still don't have any good approaches. Crystallography is not a great way of approaching this, although we have some ways. And of course, one shouldn't be one-dimensional and just be stuck to a certain technique, which we're not. But, there may not be any intermediates is another answer. And so, it's one of these things sort of like real science. It's hard to explain why something's the way it is. Probably like the economy. It's hard to explain why it is the way it is, but that's the way it is. And so, but it's not an easy problem to turn around.
Sorry, going along with that same line, I'm not so familiar with, this comes from azotobacter, right?
Yeah,
And so when you isolate this, you're isolating from that organism.
Absolutely.
So
It's not recombinant.
do you have ways of making mutants in this protein?
Yeah, so that was another thing early on, sort of in this period. There were really 3 sort of possibilities. The one that was sort of favored by the dominant group was to move all these genes into E. coli and get E. coli to fix nitrogen. That could probably work, but it didn't at that time. Another was to work on a system like Klebsiella where there was a genetic system. To me, that seems like a perfectly good approach, but no one really pursued that. The third was Dennis Dean made Azotobacter into a genetic system. And so it's not nearly as easy as E. coli, and so we don't actually make mutants, mainly because we seem to have our hands full of just the wild type protein. But it's definitely something now, you can make mutants in Azotobacter, and they've got all the proper biosynthetic machinery to make all these clusters. So that's the preferred. Yeah, no, it's a great system. And there's, whether it's the cluster assembly, regulation, mechanism, there are many, I'd say, really interesting problems, and it's not a crowded field. So, anyway.
So that sounds like an invitation for students to take this up, and with that, I'd like you to thank our speaker and congratulate our students again. Right, so that concludes today's event. You can go or come and talk to the speaker if you'd like.
Yeah, they fell out of his pocket.